Almost all eukaryotic cells have mitochondria. There may be one very large mitochondrion or hundreds to thousands of individual mitochondria, depending on what the cell needs to do. In structure, it has two membranes. It has a very smooth outer membrane and a highly folded inner membrane called cristae. The space in between the membranes is called the intermembrane space, and certain chemical reactions of cellular respiration will occur there. And inside the inner membrane is the matrix. In the matrix, you'll find DNA, ribosomes, and enzymes that all belong to the mitochondria. In elementary or middle school, you're probably told the mitochondria is the powerhouse of the cell. It is the site of cellular respiration. It produces ATP from the macromolecules. We're going to learn it mainly from using sugar, but it can use others. It requires oxygen to do that, so it's an aerobic respiration. But this organelle is semi-autonomous, like the chloroplast, as we'll see in a few moments, so it's able to move, change its shape, divide independently, and has its own DNA, so it can make its own proteins, and so that's very important for later. Plastids are a major type of organelle found in plant cells and algae, and they have a wide variety of functions. They all came about from proplastids, which are undifferentiated plastids. There are many types of plastid, not just the chloroplast that you're used to. For example, the amyloplast stores starch because amylose is a name for a type of sugar. Chromoplasts store pigments. Proteinoplasts obviously store proteins. The chloroplast is arguably the most important plastid. Like the mitochondria, the chloroplast has two membranes, but the inner membrane is just as smooth as the outer membrane. In this case, we have thylakoids inside the inner membrane, which that fluid that fills it up is not called cytoplasm like it would be in the cell, it's called stroma. The thylakoids are small discs where photosynthesis will occur, and stacks of those discs are called grana. And then they're all connected together so that they can exchange materials and resources. Like the mitochondria, it has its own DNA, ribosomes, and enzymes, and its main job is photosynthesis. Now, mitochondria had the cristae, chloroplasts have the thylakoids. Why? Well, they are both responsible for doing some major chemical reactions, and for a cell to do that kind of chemical reaction, it requires a great deal of surface area. So the highly folded cristae and the membranes of the thylakoids will give me the surface area I need to do so. Proteins that make up the fibers are very similar in all living things, from bacteria to humans. Means that they're both ancient and essential for life. The cytoskeleton is an intracellular matrix that supports cell shape and function. It's dynamic. It's composed of three main protein types, which are capable of rapid assembly or disassembly dependent upon the cell's requirements. In all cells of all domains of life, a cytoskeleton is found. Doesn't necessarily mean that the proteins are the same, but they will be similar. Structure, function, and behavior can be very different depending on organism and cell type. And within the same cell type, the structure, behavior, and function can change through association with other proteins. The main job of the cytoskeleton is to provide support and for allow for movement, but it has other roles, including regulation and homeostasis and signaling. Microtubules are the thickest of the three types of cytoskeleton components, and they are hollow, unlike the other two. Constructed of the protein tubulin, and the main job is for movement. They are what makes up your cilia and flagella. They are what makes up the centrioles inside your cells, which move your chromosomes during cell division. They also provide tracks or highways that motor proteins, as we can see on the left, can walk along to carry vesicles and other organelles. Centrioles are the MTOCs in animal cells, or the microtubule organizing centers. They create fibers that reach out and grab the centers of the chromosomes and move them around as necessary in cell division, as we will see in a later unit. But microtubules also make up your cilia and flagella, which are extensions of that cytoskeleton. Your cilia are the short ones, and you're going to have lots of them, and they move back and forth like an oar. Whereas the flagella, one, maybe two per cell, long and more undulatory, like a wave. So these things are going to allow for movement. Now, prokaryotes and eukaryotes can both have flagella, but they have an entirely different structure. So eukaryotic flagella have microtubule-based structures, whereas prokaryotic flagella do not. The intermediate filaments are, of course, intermediate in size. They're not made of just one particular protein like the microtubules. There's more than 60 different building blocks here, but the most common is keratin, the same one that makes up your hair. The job of the intermediate filaments is to deal with tension. They are deformable, they are bendable, and they can stretch up to twice their length without breaking. These are going to be permanent fixtures of the cytoskeleton that are going to keep cell shape and organelle location in place. It's going to be that skeleton, that framework that holds things where it needs to be. The microfilaments are the thinnest, and they are made of a twisted double chain of the protein actin. You might have heard of actin before because actin works with another protein, myosin, to allow for muscle movement. It's very flexible, but its main job is to assist with movement. It's also seen in cell signaling as a scaffolding protein, but you won't really see that again until AP biology. Intercellular junctions are the connections between cells. When cells become tissues, there have to be connections. There are two overall main types: open junctions, which allow the flow of materials, and closed junctions, which connect things. The two types of open junctions that we're going to discuss, one is in animal cells, the gap junction. So small molecules and electrical signals in one cell can pass through gap junctions to adjacent cells, and it's going to allow tissues to coordinate response to stimuli. For example, there needs to be a coordinated movement of muscles leading to childbirth, and gap junctions allow that to occur. The similar structure in plants is the plasmodesmata, which you can see is an opening in the cell wall between plants, allowing for the streaming of materials. Plant cells do not have gap junctions, they have plasmodesmata. The closed junctions are different. There's no opening here. The desmosome is going to be like a cellular rivet. It's going to link cells, is going to provide a connection between the intermediate filaments of the cell cytoskeletons, and it's going to give strength to your tissues. The tight junctions are your waterproof seals. It prevents materials from passing between cells. For example, the cells that line the intestine are responsible for taking up nutrients from the gut. The nature of the material taken up must be controlled to block unwanted substances from entering our bodies. The control comes from the fact that the materials must pass through receptors on the epithelial cells, but the tight junctions are going to keep things sealed. Because multicellularity evolved independently in different multicellular lineages, the composition of the extracellular matrix varies between structures. However, cell adhesion, cell-to-cell communication, and cellular differentiation are common functions of the ECM. They're made of a variety of things. Proteins like collagen and elastin are going to allow its strength and flexibility. Proteoglycans, which are protein sugar complexes, will allow for communication. And we will see proteoglycans again in the cell membrane section as glycoproteins. The only difference between proteoglycans and glycoproteins is the amount of material. So proteoglycans have more proteins than sugars, whereas glycoproteins have more sugars than proteins. That's really as far as we need to go. But the extracellular matrix is going to be important for a variety of things, and one that we're finding out a relatively new information is the regrowth and healing of tissues in the fetus. If a fetus is damaged, it is the job of the extracellular matrix to regrow whatever tissues are lost. The next few minutes you'll see a video giving you a look inside of the cell. This is of course a digital simulation, but we'll talk about it more in class.